Hey guys, so Connie asked me to upload the lectures that I recorded, but my lectures did not record very well. So I'm just gonna go over uh, last Friday's lecture for people who weren't able to come. So yeah, let's get started. Let's get started. There we go. What is doing? No, I just want one slide. There we go. Okay. Um, so overall, what he went over is this thing called flow cell, this flow cell technology from Illumina. He kind of just briefly talked about this stuff. I briefly talked about this. Okay, so this thing is the flow cell. These are a bunch of primers that sit on the flow cell. So these are all, there we go. Those are all primers, and they sit inside the flow cell. Yeah, and the read lengths. So basically, this is talking about the amount of times they flow new reagents in to continue the synthesis. So what they'll do is, let's get to a page where it shows it. They'll take your DNA, then they fragment it, then they adapt. They put these things at the end of them called adapters, and these sequences are known, and they bind to the surface of the flow cell. So basically, because all the ends here are random, what they wanted to do is be able to attach something to them that they know will attach to their flow cell. So here you got the flow cell, and this right here, this little gray part, is the primer it's binding to. And so the double-stranded DNA will bind to each of these primers. As you can see, one part of the double-stranded DNA binded to one primer, the other bound to the other primer. So this is one end, and this is the other end, I believe. Maybe this isn't the primer. Maybe this is actually just this end right here on the opposite side. Anyway, somehow this binds to the flow cell through the use of those primers I was talking about. Yeah. I'll talk about single and paired ends later. When I erase these pen marks. Anyway. So yeah. You have the lawn of primers that hybridize specific anchor sequences on the library adapters. Yep. During the sequencing run, reagents are flowing through the chambers after each cycle. Yep. Oh, so the amount of times they flow the reagents through this thing is the amount of times that, this is your read length. And it used to be where, there's a slide here somewhere. The longer your read length, I'll come back to this, don't worry. The longer your read length, I might as well just get out of the PowerPoint, find it manually. No, I don't want to keep it. There it is. Yeah, and the longer your read length, the worse your quality score gets. If you remember the ASCII conversion to the quality score thing and the SAM files, well, this is basically what this is showing on the vertical axis. These right here are quality scores, and the maximum is a 40, so I don't know why it goes up to 44. I guess it's just to look nice. So here the average is 38, but these show the ranges of how far it goes. So it starts off all these Illumina. This is all about Illumina flow cells stuff, because it's the main thing that's out on the market right now. So you have So yeah, you have this is the beginning of the sequencing, and this is towards the end of the sequencing. And as you can see, the reliability of the nucleotides at the end gets worse. But that was like 2 to 3 years ago, and our lecturer, Dr. Xiaoping Su, said that the more recent versions don't do that anymore. But I guess he wanted us to know that it used to do that. Let's see. I guess I'm just gonna stay in this version until I need to take notes for y'all, like draw on it, because I'm just gonna be jumping around back and forth, recalling whatever I can. Yeah, I guess it did. I mean, we've already kind of gone over this before with in Marco Lung's lecture, or however you pronounce his name. Yeah, you prepare the samples, you make the libraries by attaching the adapters to the ends, then you sequence them, you sequence them, and then you have all these images, and all the images need to be converted into sequences, and after that's done, you need to what's the word? Align it to a reference sequence, and then you review. Then something annotates whatever mutations you have, and then you get a review. So this part is the most demanding nowadays, because all of this stuff is easy. Making data is the easy part now. It's the analyzation of it that's hard. Excuse me. Had a bit of a post-dinner hiccup there. He didn't talk much about mate pair sequencing, so I would just kind of ignore this thing. Single or paired end libraries. Yeah, he talked a lot about paired end. That was his focus for sure. He mentioned single a couple of times, but it wasn't of dire importance. So yeah. So this right here, you can imagine this as the thing that binds to the flow cell. So you have this thing that binds to the flow cell right here, and maybe I should go into the presentation view and then do the switch view thingy. There we go. And then get my pen. Okay. Yeah. So this is the end that's bound to the flow cell. And then this end is sticking out into the solution. And so a primer adapts to this side that they already know, and it starts the PCR reaction right here. And as it goes down, it sequences whatever it synthesizes using the camera. And the camera is really high resolutions, and there's tons and tons and tons and tons and tons of these primers and these chunks attached to the surface of the flow cell. And so what a paired end read is, you have a double-stranded fragment, and it reaches the flow cell, right? So now one part attaches, and then the next step that happens is that this part right here kind of bends over and attaches to more primers on the other side. So you get something like this. And so after this happens, these strands are separated. So now you just have single-stranded things sticking out from the flow cell on separate ends, and they're opposite of each other. So a paired end would be where you read, so let's see if it binds to, I'm kind of figuring it out as I'm saying it, so excuse my slowness, but let's see. Yeah. So only this part binds, so this part will release, because this part wants to bind here. Yeah, that's how it works. Okay, so you only ligate a primer to one end of each side. So we go back here, you can see um, the ligated thing only attaches to one strand on each side. And then each of those attaches to the flow cell. So it forces the DNA to bend over like this. Then when they denature it, they come apart and they're single. So by the time you've denatured all of them, they'll all just look like a bunch of little things sticking out from the flow cell. And I don't know, I guess this right here is what this is the part that's sequenced. I just don't, maybe they're just random primers that attach here so that the reaction can start. Anyway, he didn't really go over that in detail, so maybe we can ask him next time. So your synthesis will start here, and at the same time you'll have the fluorescent thingies starting, and the camera takes pictures, and they washes away all the stuff. Then it sequences the next part, and then it flashes or whatever, makes a light, washes all the stuff away. And I think it washes the fluorophores after each step so that you don't count the old ones that you had. Right. I'm telling you, I'm really actually kind of figuring these out as I go, because I had like a vague idea of how it worked, and I actually have to tell you exactly how it works, and I actually have to go through the details on my own too. Okay, so this is the paired end read. So I'm guessing that paired end just means it ligates. They ligate primers to both sides of the DNA, like here. Whereas single read, they only ligate it to one side. And I guess there are advantages and disadvantages to each, but he emphasized paired end because it has certain advantages that aren't immediately apparent unless you go through the entire process. Well, I guess here you have it's what it's used for. Okay. Who knows? When he makes his test questions, he might ask about single end or mate pairs. But since we have him for the next 2 weeks or 3 weeks, we can ask him what he's going to put on the test. So this is his little animation. And yeah, that's another thing. You would intuitively think that they're going to read this whole section, right? So this part binds right here, and you think that, oh yeah, they're going to read this whole part up until the adapter. That isn't what happens, and there's a reason for that. So this part right here is what's actually sequenced. It's only a very small part. And you think, oh, well, shouldn't they sequence the whole thing? Because then they're going to miss out on information. Not really, because you have so many fragments that later on when you align all of the parts that you sequenced, you will be able to tell you'll be able to piece together back your sequence. And yeah. So here's the first read, and this is the second read. And all the reads on paired, this is why they're called paired, because they're given very similar IDs. So basically, the flow cell is divided into a gazillion little squares on a huge grid. And each of these squares has its own X and Y coordinate. And so you can have up to two reads that have the same coordinate. So he goes up on this later. Yeah, I'm kind of skipping over this stuff. Yeah, here we go. So this is the ID that I was talking about. And so this is for read 1. And this is for read 2. Yeah, this is think this is SAM. No, FASTQ. FASTQ is later converted into SAM, which is later converted to BAM. So FASTQ, he also gave us the reason why there's a plus here. But basically, you remember how it is. This is the ID for the read. This is the sequence. And this is the quality. And you can go to the table later and figure out what Z and U and all that means. So yeah. And then he talks about what each of these numbers means, maybe in the next slide. Oops. Crossed it out. Oh, well, there you go. Exactly what I said. And yeah, so the plus is basically a spacer. That's all it really does. But he said that the original, not the original, but, you know, when this was first made, it's still there. I mean, you should have, okay, I'm going to restart that. Basically, this is a spacer. It usually will house the ID again or something, but if you want to save space, then, like on your hard drive, then you put a plus there instead, because having just these characters repeating over and over and over again over millions and millions of reads takes up a lot of space, even though they're just characters. So not only does it save space, but I mean, it actually is a spacer. Okay, so yeah, this is what I was talking about. It will repeat the ID there, unless you option for it to be taken now and just leave this in. Okay. Yeah, so this is just part of the thing. It's not very complicated. The image basically tells everything. Every time you have, well, you have like a huge stack of images in the computer, of course, and each one is converted into sequence data for each of the pixels. So each pixel here is a DNA strand sticking out with a fluorophore shining for that flow. That's what's called a flow cell, because they flow the reagents across the primary line. Apparently this is interesting because Andrew made a noise when he saw this. So I guess it's probably because since blue light is more intense, it has to be corrected to kind of match these other ones. Let's see. Not to poke fun at you, Andrew, but, you know, whenever you say, I kind of take a second look. Let's see. Oh, this is just the base quality again. Yeah, so the base quality relates to how sure are you that this is correct, Pierre. Yeah, apparently the quality scores actually originated with Sanger sequencing, and they were carried over to this. Kind of interesting. I thought that it was only used in NGS. Okay, I think it was just a reference. Yeah, this is him just going over Sanger sequencing, I guess, and how you would, I guess, differentiate between calling it a confidently or not. So something like this you'd call confidently. Something like this you would not call because of all these dye blobs. Yeah, I guess you can just read this slide. Usually when he has text like this on his lectures, it's pretty self-explanatory. I already went over this. This is the ASCI table again. So this is for the quality scores, and the reason why they converted all of these digits to these is because these will take up 2 spaces if the character is 1 1. So they convert that into, I don't know, I guess that's a period. And so this takes up less space on your hard drive, twice as much less space, if you don't count this. Okay, continuing. Yeah, so that, like that one slide that had a bunch of rectangles going to one step to the next, this is just more detailed. Right, demultiplexing. The thing about the flow cell is that each of the lanes does not have to be specific to one sample. You can put as many samples as you want, as long as it doesn't, take up all of the primers. So you can have, depending on what you're sequencing, if you're just sequencing an exome, he said he threw out numbers like 25 to 100 samples per lane, and there's 8 lanes. That was way more than 8 lanes, just whatever. You know what I'm talking about. So he said the throughput for these machines can be really, really high. And, well, they are. This is where it goes over the nomenclature. So, instrument ID. This is just the instrument ID. I guess it's serial number or whatever they gave it for the, whatever name they gave it in the lab. Run ID, self-explanatory. Lane, self-explanatory, has 8 lanes on the flow cell. 1, 2, 3, 4, 5, 6, 7, 8. Maybe that was 8. Then you have the tile. Tile. What was tile? If I don't remember, it might have not been that important. Watch it be super important. X coordinate. It's just, I guess, if you could split up the flow cell into an X and a Y. I'm kind of guessing on this. I'm not sure if this is exactly how it is, but you get the gist. So each read has a different X and Y coordinate, and each X and Y coordinate can have up to 2 reads. So this right here is going through all the reads that are number 1, and then later, I guess it would go through all the reads that are number 2. Yeah. So IL10, 2732, 2732, 4, 4, 25, 25, 1782, 1782, 1896, 1896, 1, 1. Yeah, pretty easy. During library conditions, for its size of the layer can be selected by gel extraction and irrigation. Man, I think I zoned out on this slide. I'm sorry. I don't remember it very well. Yeah, if you want to like leave a comment or something in the Facebook comments or something, that'd be great. If this is important. Short reads. I guess this is self-explanatory. He said that long reads are better than short reads. But at the same time, he says that 75 or 76 is kind of a good zone to be in, because it's between a short read and a long read. He said the long reads are good, but they're expensive. The exact mechanism as to why, I can't really say. I think I zoned out here too. I think I was trying to fix something in in Cam Studio, which was my other recording software that I was using, which totally bugged out on me. So I don't remember anything from here. So I guess read this on your own. Alright, we went over this before, and I'm assuming he's correct, so like last time where these are reversed. I mean, it makes sense. Depth because you're going up and down, and breadth because you're going side to side. Let's hope I'm not wrong. Right, and this is him just explaining that your average coverage depth for any one base pair will be 62. So I guess that's percentages on the side right there. That says 3% of the time it'll be 62. That's the highest percentage. Well, 3.1 or whatever it is on the top here. And then 60, it'll be exactly 3%. And then, or not 60, like 63. A 90x coverage will be, only 0.2% of the time. These are just rough estimates based on the x and y coordinates here. X coverage of 10 will be nearly 0. So you have a kind of a good reliability that any base pair you run into will have a 60x coverage. Though there is some chance, of course, that your base pair is only covered 10 times or maybe even 0. Because, well, I mean, look at that. He didn't explain it, this thing here, but yeah, totally just drew over it. But you know what I'm talking about. I don't know. Oh yeah, this is just to save space on your hard drive. A lot of the chunks that you chop up from your genomic sequence will be duplicates, so you don't want that, so you get rid of them. Especially if they're very, very, very similar. Like, even though, these guys are only differing by one base pair at the end here, they're practically identical, so it gets reduced down to just... So all this will just turn into like one line, and then they'll just look like a little stack going from here to here as it goes through the genome. Might have zoned out on this slide too, so read on your own. Error rate is high in the first 1 and 2 bases. Yeah, I talked about the error rate earlier. Yeah, I mean, basically says whatever you need to know. Not sure what this is. Kind of forgot. So if you want to answer this question in the comments, go right ahead. Data processing pipeline. Yeah. Sounds about right. This is pretty self-explanatory. But I think I've already given you the overall idea of how the flow cell works. I'm just gonna kind of breeze through these last slides here and see if there's anything else I need to say. I know there is something about paired reads, the advantages of them. Yeah, this is pretty self-explanatory too. Human genome is easier to reference against because we have is that what this is about? Well, I know he said that it's harder to do de novo with NGS because you don't have a reference already. I guess these would be challenges, huh? Okay, well, basically, you can read this too. Wow. So that tells you how big the files are. Just one run is 600 gigabytes. Pretty impressive. And, it's not because the human genome is that much. It's because you have so many fragments that are overlapping each other. You have a bunch of redundant data. Oh, yeah, this is the important side. He said that the alignment, even though he doesn't talk about it much in this lecture, alignment is probably one of the most important and time-consuming parts of next-gen sequencing. And he says that Mosaic is the most robust for a whole genome, but there's basically a ton of programs out there that will align your sequence to the human reference genome. Remember, we used the BLAT last semester. Bowtie and BWA. This is, I think, what Baylor uses, he said. Bowtie, that's what Marco uses. So everyone's got their own preferences. All of them have their own little things, but the one thing I take away from all the stuff he said is that Mosaic is the one he uses, and he likes it, and he thinks it's the best. One of the things that it uses, he brought up two terms that aren't anywhere in this lecture. One of them was time complexity. And in layman's terms, this is the amount of time that it takes to... So take your information or data, put it through the algorithm, which can be anything, so we'll just make it a box here. So, imagine like getting cotton, putting it through the factory, and out comes a t-shirt. But instead you do this with info. So info goes in, algorithm churns it, and out comes process data, which here would be a bunch of unaligned unaligned fragments or unaligned reads. Algorithm aligns it to something, and then you get an aligned genome or exome or whatever you're sequencing. And this black box here is what the algorithm is. And here he referred to a thing called hash tables. And what a hash table looks like is, for one part of I kind of don't understand it completely myself either, but from what I could get was that a hash table is basically, here you have a part of the human reference genome, and he said something that was like 15 base pairs long, right? And you assign it 1, and then you reference whatever fragment you have to this table. Excuse me. And so, for instance, you have a fragment, and it's 75 BP long, and you'll reference every 15 base pairs of it to this table. If it matches one of these on here, you will assign that piece of this read, 25. And then you assign this next piece, whatever else is in the table. So if this piece is 26, your chances that this is the right part of the human genome starts to increase. And so then you take the next part, and it happens to be 27. Next part happens to be 28. So now you have a very good confidence that this chunk of DNA belongs to whatever the hash table tells you it is. And this hash table is millions and millions of numbers long. So this goes on for a long time. Probably more than me and you can fathom. With our primate brains and all. Computer does all this for us. And making this hash table is what takes up most of the time. And since you only have to make it once, that's good. He said that it takes about 4 days to make the hash table. So this is called a hash table. Every time he says it, it reminds me of hash browns. So, yeah. Mosaic uses hash tables as the algorithm to produce an aligned sequence. Done. Okay. Yeah. So he talks about mismatches. How many mismatches can you accept in all of the, I guess, all of this alignment that goes on here? And he says that 5 to 10% is okay. So this seems like it's high. But if you don't count any mismatches, you're not counting in mutations, I'd say, because you're aligning this to a reference sequence, and from human to human, we have a bunch of SNPs and all kinds of fun stuff going on. So if you had absolutely no mismatch included, you would probably never be able to align it, because no two humans are the same unless they're twins. Even then, they probably have different mutations from the environment. If they're old. From the raw sequences. Yeah. I guess the rest of this is self-explanatory. So yeah, remember he talked about the SAM format. So the FASTQ, once it's aligned, oops, that should be like that. Aligned, your computer pops out a SAM format. And SAM format is good for humans, but computers, not so much. SAMs take up a lot of space. A lot of space. And so this thing called the cigar string will hold your most important information, which is what were the mismatches. That's where your mutations are. I think that's pretty much what he went over here. Everything else is just like, where's the chromosome? I mean, for whatever mutation or mismatch you have. The chromosome, I guess, is the cigar string, tells you something about the mismatch itself. Yeah, he didn't go over about much of this. He just said, remember, this isn't the most important part. I guess we can ask him more about, why these other numbers are important. Maybe this is the reference position. Reference sequence, that makes sense. Flag, not sure. Read name, that's just given to each individual thing on the grid. Map quality, 255. We went up to 40, so not sure what that is either. Probably not important. This is probably not too important either, but I can't tell you what it is now either. Alright, so you convert SAM, which is friendly for humans, into BAM, which is binary. So you're basically taking all this info that takes up this much space, and the same exact info will now take up this much space on your hard drive. This is a metaphor. There aren't actual squares in your hard drive. As this is showing that one software we had to download, IGV. He said it's one of the best ones out there for aligning stuff. And he said he'll give us a BAM file next week so we can mess around with this. And this is just what it looks like, I guess. Yeah. Oh, well, IGV. He kind of skipped over a lot of these. Not skipped over, but went through them a lot quicker than the other ones. Here he just said what a paired read sequence can detect. So if you remember, the fragments aren't read completely. Only like this chunk of this strand and this chunk of this strand are read, and then the middle stuff is just excluded. There's a name for this middle section. I just remember what it is. Yeah, I think it's my phone, but I'll get it later. So, yeah. Okay, so yeah, this refers to this. This refers to that. These lines refer to this gray part right here. So yeah, that tells you if there's a point mutation or not, because I guess these don't have anything. Usually they like write, like, oh, sorry. Wow. Smart me. Yeah, this is the reference sequence, and then all of your reads have a C instead of an A, so point mutation. And then your reference sequence has something here, whereas your aligned stuff has nothing there, so deletion. And see, it's not deleted for all alleles. Some of them still have it, so there's that. Homozygous deletion, everything's gone. There's not one thing that has it in your entire whatever they extracted it from. But here, only one of your alleles got taken out. Because compared to everything else that you have like an average of this many, I'd say. I'm totally kind of guessing right now, but I'm kind of sure that I'm right. So if you have an average amount of maybe this many reads per area, and you come across where you have half as many reads for a certain area, it's probably why homozygous deletion. Yeah, copy number alterations. Yeah, so I guess this would be in reference to how many average reads you have. So if something is half as many as all the others, and it's very consistent, then you check that out. If it's twice as many, you check that out. And I'd say this stuff right here, this is probably all much more easily done with CGT stuff, but I guess it's just saying that you can detect these things with NGS. And yeah, here's a translocation like the Philadelphia chromosome, or like, what was it, 15, 17, 9 something, 9, 10, 11, I don't remember. But yeah, going on. Yeah, self-explanatory. He just completely skipped this slide. That I remember well. Targeted sequencing. He started talking about microRNA sequencing here, saying that you could or couldn't do it. I think he said you could do it, because microRNAs are only like 7 to something small base pairs, 7 to 30. It was something really small. That's all I remember. It's kind of fuzzy right now. So I guess this is where he starts talking about RNA sequencing. Hey. He did say something about this, but I don't remember it at all. I mean, he didn't spend a large amount of time on this, I'll just say that. Oops. Okay, ignore that. Continuing. I just went up. I guess this is so I guess this would be kind of like how we had real-time PCR, where we compared a reference housekeeping gene to whatever gene we were supposed to have, whatever gene we were targeting, and we'd see if their expression level was increased or not. Something like that. So this is saying, hey, we had this many. So they're here not doing DNA on the flow cell, but they're using RNA. That's supposed to be a flow cell. There you go. And they see how many copies of every RNA thing do we have. And depending on how many copies they have, because based on random chance, the ones that are higher frequency in the cells will more likely bind to the lawn of primers, and you'll get more depth of coverage for those areas. So this is like, I guess each one of these peaks is a different base pair or set of base pairs. I don't remember much about this slide at all. He did go over this for a little while. I think based. Oh dear, what just happened? Okay, based on this stuff, you can basically make a microarray and track gene expression. And yeah, so decreased expression, increased expression. And then you guess each one of these dots counts for a different gene or some going on. Or RNA. Yeah, RNA. Cryptome sequencing to detect gene fusions. Yeah, I was talking about this earlier. Oh yeah, and the reason you need paired reads, I forgot to talk about the advantages of paired reads, is that paired reads come from the same fragment. So this part of one read is kind of matched to this part of this read, and if you remember, they start out like this. So I'm assuming, however they're given their IDs, that they're right next to each other. That since if you have, for instance, this part of the genome right here that is unique, but on the other strand, this part of the genome is matched to a bunch of other genes or pseudogenes or repeat sequences, then what you can do is look at all the different places in the genome that have this sequence, and based on how far away these two fragments were, you can say, oh, so if this is unique and this is matched to a bunch of other places, then whatever is this far away from this is probably where this part of the sequence belongs. And if both of these sections are not unique, well, you have to throw both of them out because you can't confidently pair them anywhere. But if one of these is unique and the other one isn't, you can, with some degree of confidence, say that because it has an ID that relates to this and it's yay far away, that it's this is what got sequenced for this section of the genome. I hope you got that. So remember that if you have one read going this way and the other read going this way, that there's a gap in between the reads, and so this will sequence this way and this will sequence this way. And there was a slide that showed you the IGV. So you see the reason they have these things is because when you have paired reads, excuse me, so you'll sequence it this way, so it'll go forward, and if you sequence it this way, it'll go backward. So as you can tell, I mean, there's not a whole one here, but there's a blunt end and a little hat end, and this tells you which direction it was sequenced in. Hopefully you got something out of that. Yeah. Not sure about this part. Might have skipped over it. He might have only talked about it for, 30 seconds or so. Awesome. I guess read over that. And, the $100,000 analysis, $1,000 genome thing. This is an assignment did in class. We had to turn it in. If you haven't turned it in, I'd recommend emailing Crystal. And that's it. No. Hopefully you found that helpful. I'll see y'all on Thursday. Hopefully the ice doesn't get us again.